Cloning Cry1Ac by using E. coli. The contents we will talk about today will start off with an introduction along with the cloning process, followed by the separation and purification process, then the applications, and ending with the overview and perspective. So, what is the purpose of cloning genes on a large scale? The purpose of cloning genes on a large scale is to produce a significant quantity of a desirable gene or the protein it encodes for various applications including research, biotechnology, and pharmaceutical production. Why do we use E. coli to clone genes? E. coli is the most frequently used host for the production of enzymes and other proteins by recombinant DNA technology. It is commonly used for gene cloning because it is easy to culture, genetically manipulable, compatible with plasmids, has efficient transformation, and offers standardized tools and safety in laboratory settings. So what exactly are we cloning? We are cloning the Cry1Ac gene. The Cry1Ac gene is an example of a gene that has been cloned in E. coli. The gene comes from Bacillus thuringiensis, a soil bacterium commonly used as bioinsecticide, which is also known as Bt. The protein toxin encoded by the Cry1Ac gene synthesizes crystalline cry protein, which is toxic and causes pathogenicity against mainly three insect orders, Coleoptera, Diptera, and Lepidoptera. Some common insect species include the tobacco budworm and cotton bollworm. The importance of cloning the Cry1Ac gene in E. coli is for the large-scale production of the Cry1Ac protein in order for usage in field crops, which will be further explained in the upcoming sections. Next, we will explain the cloning process of the Cry1Ac gene from Bt using E. coli. There are roughly 5 steps in Cry1Ac cloning. First, isolating the gene. Second, generating a cloning vector. Third, inserting the gene into the vector. Fourth, transformation into E. coli. And fifth, selection and culturing. To start off, the cloning of the selected gene using E. coli is initiated by isolating the gene from its original state. The Cry1Ac gene can be obtained by identifying and isolating the specific DNA sequence from Bt by culturing the Bt strain in a suitable medium. Then, bacterial cells are harvested at the desired growth phase, which is usually the mid-log phase, and we perform cell lysis to release genomic DNA. After that, we purify the genomic DNA using a phenylchloroform extraction to get the Cry1Ac gene. Next is the second step, generating the cloning vector. Cloning vectors are DNA molecules into which foreign DNA can be inserted. Typically, scientists adapt naturally occurring structures that can replicate independent of chromosomal DNA. For Cry1Ac cloning, shuttle vectors such as PHB201, PHSP9, PEG597, and PEG853 were used as they are designed with rare restriction sites that permit various adaptations, including the construction of small recombinant plasmids lacking antibiotic-resistant genes. The plasmid is then prepared with appropriate restriction enzymes called restriction endonucleases, which is necessary as it provides a system that will ensure that the cell's own DNA is not degraded. This protection is usually provided by the modification of the DNA, such that the sites recognized by the endonuclease are methylated. They also act as molecular scissors to cut out the desired gene on the restriction sites and contain antibiotic-resistant markers. This vector will carry the Cry1Ac gene. Then we come to the third step, inserting the gene into the vector. The Cry1Ac gene is inserted into the cloning vector using the same or compatible restriction enzymes, creating compatible sticky ends on both the Cry1Ac gene and the vector. The sticky ends are then ligated together using DNA ligase to create the covalent bonding of the vector and the Cry1Ac gene to create a recombinant DNA molecule. The fourth step is the transformation into E. coli. The recombinant cloning vector is introduced into E. coli cells using transformation techniques such as heat shock, electroporation, or chemical transformation. After transformation, the cells that have taken up the vector can be selected based on their antibiotic-resistant markers on the vector. The fifth step involves selection and culturing. The transformed E. coli cells that have taken up the Cry1Ac genes are then selected and cultured on agar plates containing the antibiotics corresponding to the resistant genes on the vector. Then, they are incubated the plates overnight at the appropriate temperature, which is a suitable environment for cells to grow into colonies. That temperature is usually around 37 degrees Celsius. The selected colonies are further cultured in liquid culture for plasmid extraction. In the end, successful cloning is confirmed by performing techniques such as PCR, DNA sequencing, or restriction enzyme digestion to analyze and verify the presence and integrity of the cloned Cry1Ac gene in E. coli.
Come to the separation and purification part. After cloning the Cry1Ac genes in E. coli, the next step would be to separate and purify the Cry1Ac proteins from the transformed E. coli cells. Here is the general process for putting the Cry1Ac proteins back into the new produced and bioinsecticide. In protein separation process, the E. coli cells containing the cloned Cry1Ac genes are grown in a culture medium like bioreactor, which is a manufactured device that supports a biologically active environment for cells to exponentially grow. Once the proteins are expressed, the cells are harvested, and the Cry1Ac proteins are separated using cell disruption techniques, such as sonication that apply sound energy to agitate until cells break apart, or French press homogenizer that cells are forced through the tiny orifice and rupture into solution that contains cell contents. Then the solution is thrown to a microfilter and centrifuged to remove most of the contamination and become a clarified liquid called lysate. But it is not purified enough, so we require purification process. In this process, the lysate is purified to obtain highly concentrated and pure Cry1Ac proteins by various chromatographic techniques, such as affinity chromatography, by using the advantage of the unique binding property of the target molecule to specific ligand. Size exclusion chromatography, the separation molecule based on their size by filtration through a gel, which occurs when molecules of different size are included or excluded from the pores within the matrix. Ion exchange chromatography, which relies on the reversible exchange of ions between the molecules in the sample and the charged groups on the stationary phase. The hydrophobic interaction chromatography, by taking advantage of the hydrophobic interaction based on difference in their hydrophobicity. These techniques are commonly used for highly specific protein purification to ensure that unwanted impurities or contaminants are removed. To confirm the identity and purity of the expressed protein through the technique like mass spectrometry, for measuring the mass to charge ratio, which is used to calculate the exact molecular weight of the sample components. SDS page, which is an electrophoresis method that allows protein separation by mass. Lastly, isoelectric focusing. It is a technique in electrophoresis that separates proteins or molecules based on their isoelectric point. We can combine SDS page and isoelectric focusing to get both results. To store the purified Cry1Ac proteins in good condition, the stabilization process is required. It's typically stabilized to improve their stability and ensure their efficacy as bioinsecticide. This may involve adding stabilizing agents or formulating the proteins into a suitable delivery system, such as encapsulation in microsphere, or mixing with adjuvant substance. Now, the application to new produce. The formulated Cry1Ac protein can be applied to new produce in several ways. First, spraying. The Cry1Ac proteins can be diluted in water or a suitable carrier to solution and sprayed onto the produce, targeted the pest that bioinsecticide is effective against. Secondly, coating or dipping. The produce can be coated or dipped in a solution containing the Cry1Ac proteins, allowing the proteins to adhere to the surface and provide protection against pests. Third, genetic modification. In some cases, the Cry1Ac gene can be introduced directly into the plant's genome to enable the plant to produce the desired Cry1Ac protein. This method is known as genetically modified organism technology, or GMO. In overall perspective, on production scale, the advantage of cloning by using E. coli is because it allows larger scale production and provides sufficient quantities of Cry1Ac protein for various commercial BT products. In function, once the Cry1Ac protein are applied to the product, the protein binds to specific receptors in the guts of susceptible insect larvae, disturbing their digestive system and ultimately leading to their death. About the environmental concern, the use of Cry1Ac protein as bioinsecticide offers an environmental friendly and non-harmful to human alternative to chemical insecticide, and can help control pest population in agricultural settings.